How does your heart suddenly beat faster during stress? How do medicines for asthma open narrowed airways? And how do hormones communicate with cells throughout the body? The answer lies in one of the most important receptor systems in medicine, G-protein coupled receptors, commonly known as GPCRs. So, welcome back to our Receptors series. In this session, we will understand what GPCRs are, why they are important, how they are structured, and how they convert an external signal into a cellular response. Given the complexity and clinical importance of G-protein coupled receptors, we'll discuss them in a three-module series. In Part 3, Module A1, we'll understand the introduction, structure, and mechanism of action of GPCRs. In Module A2, we'll explore the GS, GI, and GQ signaling pathways and understand the role of second messengers, such as cyclic AMP, inositol triphosphate, DAG, and calcium. Finally, in Module A3, we'll compare GPCRs with ligand-gated ion channels, discuss major receptor examples, clinical importance, pharmacological relevance, and high-yield revision points. So, let's begin with the fundamentals of GPCRs. G-protein coupled receptors, or GPCRs, are a large family of cell surface membrane receptors that allow cells to communicate with their external environment. These receptors detect chemical signals outside the cell and convert them into intracellular responses through specialized signaling proteins called G-proteins, also known as guanine nucleotide binding proteins. In simple words, GPCRs act like molecular messengers. They receive signals from outside the cell and instruct the inside of the cell how to respond. Unlike ligand-gated ion channels, GPCRs do not directly open an ion channel. Instead, they work indirectly through intracellular signaling cascades, making their responses slower but often more amplified and long-lasting. GPCRs are known by several different names, and each name reflects an important feature of the receptor. They are called Seven-transmembrane receptors, or 7-TM receptors, because they pass through the cell membrane 7 times. They are also called serpentine receptors, because of their characteristic snake-like arrangement within the membrane. Another important term is metabotropic receptors, which means they produce effects indirectly through intracellular metabolic signaling pathways rather than directly controlling ion movement. Why are GPCRs so important? GPCRs are among the most important receptor systems in the human body because they regulate an enormous variety of physiological functions. In the nervous system, GPCRs help regulate mood, learning, memory, behavior, neurotransmission, and pain sensation. Neurotransmitters such as dopamine, serotonin, norepinephrine, and muscarinic acetylcholine exert many of their effects through GPCRs. In the cardiovascular system, GPCRs regulate heart rate, blood pressure, vascular tone, and cardiac contractility. For example, stimulation of beta-1 adrenergic receptors increases heart rate and force of contraction, whereas M2 muscarinic receptors slow the heart. In the respiratory system, GPCRs regulate airway smooth muscle tone. Activation of beta-2 receptors relaxes bronchial smooth muscle and produces bronchodilation, which is why beta-2 agonists are widely used in asthma treatment. Many hormones also signal through GPCRs, including glucagon, adrenocorticotropic hormone, thyroid-stimulating hormone, and vasopressin. GPCRs also regulate gastrointestinal motility, gastric acid secretion, immune responses, and even sensory functions. In fact, our sense of smell depends heavily on olfactory GPCRs, and vision involves rhodopsin, which is another classic GPCR. From a pharmacological perspective, GPCRs are extremely important because they are among the most common drug targets in medicine. Nearly 30 to 40% of clinically used drugs act either directly or indirectly on GPCRs. Medicines targeting GPCRs are used to treat conditions such as hypertension, asthma, depression, schizophrenia, allergies, and chronic pain. For example, beta receptors are targeted in cardiovascular disease. Serotonin receptors are important in psychiatric disorders. Histamine receptors are involved in allergy treatment, and opioid receptors are targeted in pain management. This is why understanding GPCRs is absolutely fundamental in pharmacology. Before discussing structure, let us first understand some key characteristics of GPCRs. First, GPCRs form the largest receptor family in the human body, with more than 800 identified receptors. Second, GPCRs are capable of signal amplification. This means that a very small signal outside the cell can generate a massive response inside the cell. For example, a single receptor activation can stimulate multiple intracellular signaling pathways, dramatically amplifying the signal. Third, compared with ligand-gated ion channels, GPCRs usually act more slowly. While ion channels often respond within milliseconds, GPCR responses generally occur over seconds to minutes. However, GPCR-mediated effects are usually more prolonged and sustained. Finally, GPCRs work indirectly through G-proteins and second messenger systems rather than by directly opening ion channels. Now let us understand the general structure of a G-protein-coupled receptor. Although GPCRs recognize many different ligands, most share a remarkably similar structural framework. Structurally, GPCRs are single polypeptide membrane proteins that pass through the plasma membrane 7 times. This 7-transmembrane arrangement is the hallmark feature of GPCRs. To understand their structure more easily, we can divide a GPCR into 3 major regions: the extracellular domain, the transmembrane domain, and the intracellular domain. First, the extracellular domain lies outside the cell membrane and serves as the ligand binding region. This is where signaling molecules such as neurotransmitters, hormones, and drugs bind. Different GPCRs possess different ligand binding pockets, which determine receptor specificity. For example, the beta-2 adrenergic receptor recognizes epinephrine, whereas opioid receptors recognize endogenous opioids or drugs such as morphine. Second, the transmembrane domain consists of 7 alpha helical segments that repeatedly cross the membrane. These helices are connected by extracellular and intracellular loops. This region is responsible for maintaining receptor stability and transmitting signals across the membrane. When a ligand binds to the receptor, these helices rearrange their structure. This structural rearrangement is called a conformational change, and it represents the key step that initiates intracellular signaling. Third, the intracellular domain is located inside the cell and interacts directly with G-proteins. This region acts like a molecular switch, transmitting signals from the receptor to intracellular signaling proteins. Now that we understand GPCR structure, let us look at the intracellular partner that makes GPCR signaling possible: the heterotrimeric G-protein complex. GPCRs communicate with specialized intracellular proteins called heterotrimeric G-proteins, which act as molecular switches to transmit signals inside the cell. The term heterotrimeric simply means made up of 3 different subunits. These are the alpha, beta, and gamma subunits. In the resting state, these 3 subunits remain attached to one another, forming the inactive G-protein complex. Among them, the alpha subunit is especially important because it binds small molecules called guanine nucleotides, which determine whether the G-protein is active or inactive. You can think of the G-protein as an on-off molecular switch. When the alpha subunit is bound to GDP, or guanosine diphosphate, the G-protein remains inactive, or off. However, when a GPCR becomes activated, it causes GDP to be replaced by GTP, or guanosine triphosphate. Importantly, remember that GDP is not converted into GTP. Instead, this is an exchange process. Because the cytoplasm contains a much higher concentration of GTP, a GTP molecule quickly binds to the empty nucleotide binding site on the alpha subunit. This exchange acts like flipping a switch. GDP means off, while GTP means on. Once GTP binds, the G protein becomes active and undergoes a structural change. The alpha subunit separates from the beta-gamma complex, and both components can activate intracellular signaling pathways. Eventually, the signal is turned off when the alpha subunit converts GTP back to GDP, allowing the three subunits to rejoin and return to the inactive state. Now that we understand the structure of GPCRs and G proteins, let us see how GPCR signaling actually occurs step by step. Step 1. Ligand binding, signal recognition. The process begins when a signaling molecule, or ligand, binds to the extracellular region of the GPCR. These ligands may include epinephrine, acetylcholine, dopamine, histamine, hormones, or drugs. This interaction is highly selective, meaning that each receptor recognizes only certain ligands. For example, beta-adrenergic receptors recognize epinephrine, while opioid receptors recognize endogenous opioids and opioid drugs. You can think of this interaction like a lock and key mechanism, where only the correct ligand can properly activate the receptor. Step 2. Conformational change, receptor activation. Once the ligand binds, the GPCR undergoes a structural rearrangement known as a conformational change. Although the receptor remains in the membrane, its shape changes significantly. This conformational change is especially important on the intracellular side of the receptor, where it creates an activated surface capable of interacting with the G protein. In simple terms, ligand binding changes the receptor from an inactive state to an active state. Step 3. G protein activation, GDP to GTP exchange. As we already discussed under G protein activation, the activated GPCR interacts with the heterotrimeric G protein. In the resting state, the alpha subunit contains GDP and remains inactive. The receptor then stimulates the exchange of GDP for GTP, thereby activating the G protein. Remember, GDP means off, whereas GTP means on. Step 4. G protein dissociation. As mentioned earlier, activation causes the G protein to dissociate. The alpha GTP subunit separates from the beta-gamma complex, and importantly, both components can activate downstream signaling pathways. Step 5. Activation of effector proteins. The activated G protein components then interact with effector proteins located within the cell membrane. These effectors may be enzymes or ion channels. Important enzyme effectors include Adenylyl cyclase, which produces cyclic AMP, and phospholipase C, which generates signaling molecules such as inositol triphosphate and diacylglycerol. GPCRs can also regulate calcium and potassium ion channels, thereby influencing cellular excitability. Step 6. Second messenger formation, signal amplification. These intracellular pathways generate second messengers, which amplify the original signal. This amplification is one of the major reasons GPCRs are so powerful. A single activated receptor can trigger multiple intracellular signaling events, producing a much larger response inside the cell. Ultimately, these signaling pathways produce physiological effects, such as changes in heart rate, smooth muscle contraction, neurotransmission, secretion, or gene expression. Step 7. Signal termination, resetting the system. Finally, the signal must be turned off to prevent excessive stimulation. The alpha subunit possesses an intrinsic GTTase activity, meaning it can hydrolyze GTP back to GDP. When GTP is converted to GDP, the alpha subunit becomes inactive again. It then reassociates with the beta-gamma complex, restoring the inactive heterotrimeric G protein. In this way, the system resets and becomes ready for the next signal. But how exactly do these signaling pathways work? How do GS, GI, and GQ proteins produce completely different cellular responses? And what roles do second messengers such as cyclic AMP, inositol trisphosphate, diacylglycerol, DAG, and calcium play? We'll understand these mechanisms in Module A2. If you found this video helpful, don't forget to like, share, and subscribe for more simplified pharmacology and medical science content. Thank you for watching, and I'll see you in the next video.